Today's topic for discussion is life cycle of bacteriophages or reproduction of bacteriophages or multiplication of bacteriophages. Let us see the details. The bacteriophage comes in close contact with bacterial cell or E. coli cell, and the tail plate of virus comes in contact with the bacterial cell wall. The tail fibers of virus get attached to the cell wall of bacterium at a specific site that the tip of the phage tail happens to touch. Then the next step is the injection of DNA of virus into bacterial cell. Within a few seconds of attachment, a opening is formed on the bacterial cell wall at the point of attachment. The protein sheath of the tail now contracts by moving the hollow core into the host cell or bacterial cell. The viral DNA is then forced down into the bacterial cell. The viral DNA is transferred to the bacterial cell through opening. The protein coat of virus remains attached to the empty shell outside the bacterial cell. This step is also described as adsorption and penetration phase or infection phase in the life cycle of bacteriophages. And what happens then? Step 3 is the latent period phase, which is also known as period of eclipse. This period takes 12 to 22 minutes to complete. During this period, no viral DNA is seen into the infected bacterial cell. It disappears as an independent infective agent. It seems that the viral DNA within the host cell enters the chromatin body of bacterium and takes control of the cell's genetic machinery in the manner unknown at present. And the replication of bacterial DNA and synthesis of bacterial enzymes stops immediately or changes immediately, and under influence of the phage DNA, the chromatin body of the host, which also contains DNA, is broken down and its contents are dispersed within the first few minutes after infection. At this stage, the bacterial enzyme system converted into systems capable of elaborating phage components from the cell contents and ingredients absorbed from the medium. The phage DNA is replicated in the region of chromatin body of bacterium or host cell. The ribosomes of bacterial cell, the ribosomes which are present in bacterial cell, that start producing new phage coat protein, new viral protein. They produces viral proteins. And the next step in the phage life cycle is assembly of the phage components into new phage particles. In this step, the elaborating particles of bacteriophage DNA leave the chromatin region of the host cell. The new phage protein quickly aggregate around them to constitute a new stable viron of T1 phage or bacteriophage. Assembly of phage units starts about 12 to 22 minutes after infection. The tail is finally added to the phage head. Within about half an hour. Within half an hour after infection, the host cell contains 2 to 300 or even more particles of bacteriophages as shown in the photograph. Bacteriophages or T1 phages. These are also called as T1 phages. The period of production of new phage particles is called vegetative phase. It comprises the replication of DNA, synthesis of phage protein and assembly of phage component into a new phage particles or bacteriophage particles. The final step in the phage life cycle is liberation of phages or bacteriophages. The host cell bursts to open phage progeny. All the bacteriophages are released. The process of disintegration or destruction of coli phage is called lysis. The phage which brings about lysis or destruction of host bacterium is called a virulent phage. This is the multiplication of bacteriophages which multiply in the bacterial cell. Thank you very much. Save trees, save earth.